All right, can you hear me?
We can hear you, but now we can't see your screen. There we go. Now we're all set.
Excellent. Okay, we've got
Perfect.
it all
Thank you.
together.
Yeah.
Excellent. Well, thank you all so much for having me. I appreciate the introduction. So I'm not located in the Delaware Valley, but I do work very closely as a pharmacogenomic specialist at Nemours in a program that works throughout the Florida locations of Nemours and in the Delaware Valley. My primary appointment is as faculty at the University of Florida College of Pharmacy. So, in terms of what I do in my work, so my clinic is a clinical pharmacogenomics clinic that I'll talk about in a moment at Nemours Children's Health. Also on the call, I think today, one of my colleagues, Benjamin Duong, who covers the Delaware Valley portion of our clinic. So, if you have any interest in what we do from a clinical pharmacogenomics standpoint, I would encourage you to reach out to him either for clinical or
Oh, Nathan, you're muted.
Thank you so much. I don't know why it keeps muting me. So my research is focused on primarily on pediatric sarcomas, and then I also teach at the University of Florida primarily in precision medicine and oncology. So to briefly cover pharmacogenomics, I'm sure many of you have already heard about what pharmacogenomics is. Very briefly, it's the relationship between an individual's genetics and how they respond to medications. And if we can accurately predict those responses based on someone's genetics, we can help to personalize their drug therapy by identifying drug response, risk for adverse events, and determining the optimal drug and or dose. So we do this at Nemours Children's Health through the Delaware Valley and Florida locations. We have our own in-house test, but also are able to support external commercial laboratory testing. We have our own clinical decision support services that we maintain with an external vendor. We also run a consult service where we provide guidance and education for patients and for providers based off of the results from our service. In addition to our clinical work, we do also offer and perform research activities that run the gamut from discovery science, clinical science, implementation science, and quality assurance. So again, if there's any interest in pharmacogenomics, if you're interested in adding pharmacogenomics to your current work and would like to partner with us, I would encourage you to reach out either to myself or my colleague in the Delaware Valley, Benjamin Duong. We would love to expand the work that we're doing and to partner some collaborators as well. So I was primarily invited here to talk about my own personal research, which I will do now, which is not directly related to pharmacogenomics, but is more focused on precision medicine in cancer, doing what I call clinical computational pharmacology. And what that means is that my lab bridges gaps between translational science through in vitro and vivo studies and retrospective clinical studies, computational science with multi-omics and big data analysis, and then clinical science with pharmacokinetics correlates and clinical trial. My areas of interest generally are tumor heterogeneity, both within a tumor and between tumors, integrating multi-omic and clinical data, mechanisms of drug resistance, and doing correlative pharmacology. So I'll provide one example of how we combine all of these different kinds of science into meaningful contributions to the literature through a paper that we published a couple of years ago, looking at the base excision repair pathway in multiple myeloma. Multiple myeloma is an incurable plasma cell malignancy, and one of the primary ways that we treat multiple myeloma is through autologous stem cell transplant, where you retransplant a patient's own stem cells back to them after giving them an incredibly high dose of chemotherapy. In doing that, you wipe out a large portion of multiple myeloma. However, it is not a curative modality. So some multiple myeloma cells will survive to seed further disease. The response to autologous stem cell transplant then is highly variable, and a lot of it comes down to response to that really high dose of chemotherapy that we give before the transplant. It's highly variable. So we hypothesized that DNA repair capacity could be one of the ways that multiple myeloma escaped this cytotoxic chemotherapy before stem cell transplant. From some preliminary data, we assessed that the base excision repair pathway would be a leading candidate for this study. So we assessed that whole pathway across multiple data sets from patient samples ranging from normal plasma cells to intermediary states to full multiple myeloma. And what we identified was that as multiple myeloma developed, we saw an increase in expression and activity of the base excision repair pathway. From there, we were able to collect data from multiple clinical data sets to assess survival outcomes after autologous stem cell transplant based on the expression of our leading two genes from the base excision repair pathway, PARP1 and POLD2. For high expressing tumors of both of these genes, clinical outcomes were incredibly poor after autologous stem cell transplant. We also had data from patients who never received an autologous stem cell transplant. And for those patients, expression of PARP1 and POLD2 did not confer a better or worse response, suggesting that this is specific to response to the autologous stem cell transplant modality. We then brought that data back to the lab, and using a PARP inhibitor, which blocks the base excision repair pathway, we were able to identify that blocking that pathway sensitized multiple myeloma cells to the chemotherapy that we use in autologous stem cell transplant treatment for these patients. So all of that put together suggests that this pathway might be a biomarker for response to this treatment modality, and also may help us identify ways to improve the treatment modality by addition of a PARP inhibitor. So that's one example of a way that our lab approaches the science that we do. Our lab mostly focuses on sarcomas, and I'll give you two examples of some projects that we've been working on very recently here. But we do work across cancers using those tools as ways to better understand multiple aspects of tumor biology and try to translate it to a pharmacology focus as well. So what I'd like to introduce now is a couple ideas, and one is the idea of fusion-driven sarcomas. Some of you may have heard of these before, but this is an incredibly rare subset of cancers that is most frequent in pediatric populations. And these are sarcomas that are driven by a fusion gene between two transcription factors, meaning that the presence of that fusion gene completely alters the somatic transcriptomic landscape of a cell, and that fusion alone is able to drive the development of the cancer. Most of these cancers have very few mutations. Most of them will only have that driver fusion as the thing that's causing the cancer. So this is not like a melanoma or cancers that slowly mutate over time. They have this pre-malignant state before they become cancerous. These cancers, you have one alteration and it's a cancer. Because they primarily work by altering the transcriptomic landscape of the disease, it's really difficult to identify what pathways are most important to the biology of those cancers, and then how we can use that to develop better drugs to treat them. One of the things our lab has found over multiple different studies is that even though the mutation rate in these cancers is very low, if you look at the rare mutations that are secondary to that driving fusion, they can give you hints of what pathways could be most important to that cancer's oncogenicity. So one example would be looking at the ultra-rare sarcoma, which is called chick ducts translocation positive sarcomas, or CDS. These are incredibly aggressive, fast-growing cancers. They grow faster than Ewing sarcoma, which is a similar disease. They're heavily chemo-resistant, and their survival is really poor. Because they're ultra-rare, we have very limited knowledge about how these cancers work. and what we could use to potentially target them more than just using traditional chemotherapy. So what we did in a recent study that we published, we looked at multiple existing data sets, including in vitro and in vivo data, patient data, and murine-derived cell line data. And because this is an ultra-rare cancer, all of these data sets are very, very small. So we took an approach where we ran multiple experiments and looked at the overlap of signals to identify a molecular signature that was specific to CDS. We then ran that overlapping signature and compared it to every cancer genome that we could get our hands on, and then validated it in murine samples. All of this to focus down on a highly specific CDS gene signature. And what you can see here is that we ended up with a gene signature. I hope you can see my cursor. A gene signature of 5 different genes, including ETB145, DUSP4, and POLE-E, identified on this PCA plot here on the right. These 5 genes were highly expressed in chick duct tumors, or CDS tumors, as opposed to Ewing sarcoma tumors, and opposed to every other cancer type that we were able to compare with. We then validated that signature in a number of murine models, all of them demonstrating that CDS increased the expression of these 5 genes. So of those 5 genes, the ETVs and DUSP4 have actually been previously reported as highly expressed in CDS, but POLE-E has not. POLE-E is a DNA repair gene, but in cancer, it's typically deactivated. So what we're seeing here is an increase in expression of POLE-E, which is highly unique in cancer. So we then did additional analysis of the data that we had, looking at a pathway level analysis, and identified that across the DNA repair pathway spectrum, CDS had an increased expression of this DNA repair ability. And then we tied this DNA repair activity through POLE-E with the high proliferative ability of CDS. So again, the clinical profile we see is they grow very quickly, which would suggest that they should respond to chemotherapy because they are dividing so rapidly. But they're also chemotherapy resistant. So what we see here is that POLE-E tracks very closely to a measure of cellular proliferation in Ki-67. Though there was a heavy positive correlation across cancers and within CDS samples alone. So this said to us that we have this model now where the fusion in CDS is driving proliferation, but it's also driving DNA repair. And very interestingly, a couple months after we published, a different group published a paper using completely wet lab methods and identified POLE-E as a direct target of the chick duct fusion, and showed that it's essential for DNA or for retaining DNA integrity during cellular division. So this is specifically expressed with the insertion of the chick duct fusion. And if you got rid of POLE-E specifically in these cells, you decrease tumor formation and growth rate. It led to increased DNA damage and an increased P21 mediated senescence. So this actually validates our model that we came to computationally, suggesting that cell cycle activation and DNA repair go hand in hand in this extremely aggressive disease. The last example that I'll give, and I'll wrap up really quickly here, is some work that we're currently doing looking at the gene CDKN2A, which codes for the protein P16 in Ewing sarcoma. Ewing sarcoma is a bone and soft tissue malignancy that's most frequent in children. It's actually a curable malignancy. We can cure a large number of patients that have early stage disease with our upfront chemotherapy treatment modality. Chemotherapy combined with surgical removal and radiation leads to a high rate of response. But for patients whose tumors do not respond, if they come back, or if they're metastatic at presentation, we have no medications that are effective for them. So we want to understand better if response to initial chemotherapy is so important for the overall survival of these patients, how can we better identify which patients won't respond to chemotherapy, and how do we take those non-responding tumors and force them to become responsive? There are two genetic alterations that are relatively frequent in this disease, STAG2 and TP53. When they're altered, outcomes are typically worse for patients, but there's another gene that's frequently altered in Ewing sarcoma, which is CDKN2A. We don't really know what this does for the disease. There's conflicting reports about whether it confers a poor survival outcome or a neutral survival outcome. Given our previous work, we don't really believe that alterations in CDKN2A are random. So we wanted to dive deeper into figuring out why we're seeing this recurrent alteration in this gene. We recently published a paper revisiting CDKN2A in Ewing sarcoma, where we revised a lot of analysis that had previously been done, and then adjuncted that with some of our own internal data. What we first were able to demonstrate very clearly using some CRISPR knockout data was that the downstream pathway of CDKN2A is an important dependency for sarcoma. So if CDKN2A is active, it blocks CCND1 and CDK4. Cells cannot proliferate, particularly in a petri dish. Ewing sarcoma is very sensitive to activated CDKN2A. And this is why we believe that a lot of Ewing sarcomas lose CDKN2A. About 15% of them do. It gives them a growth advantage. However, we also showed that if you have very high expression of CDKN2A in Ewing sarcoma, particularly for clinical samples, their survival outcomes were poor. So how could deletion of CDKN2A, which leads to a faster growing tumor, and high expression of CDKN2A, which we would expect leads to a slower growing tumor, but also leads to poor overall survival? How do we check those two things together? Well, we believe that based off of the characteristics of how these data were collected, that CDKN2A doesn't confer a single survival direction. It's not only good or only bad, but it depends on the context. In the context of pure growth ability of a cell, losing CDKN2A is actually really good. And that's what we found in preclinical models. But if you're going to give the tumor chemotherapy, we believe that having high expression of CDKN2A allows those tumor cells to escape chemotherapy. So that was our hypothesis, and that's what we have been working on testing recently in the lab. So what we did is we developed stable CDKN2A expression models using CDKN2A null Ewing sarcoma cell lines, and then introducing an overexpression vector of CDKN2A into those cell lines. So we currently have 4 cell lines that have no expression of CDKN2A, 4 of those same cell lines where we've overexpressed CDKN2A, and we also have 4 cell lines that have endogenous but low expression of the gene as well. What we found when we compared the null expressing cells to the cells that we overexpressed CDKN2A in was that the overexpression cells obviously highly expressed P16, which is the protein CDKN2A, but that expression of P16 led to a senescence phenotype in all of our models. So high expression of CDKN2A, high expression of P16, induction of senescence. And senescence is a dormancy state of the cell. When we treated those cells with chemotherapy, those cells that were going into that senescent dormant state that had a high CDKN2A expression, those were less sensitive to our chemotherapeutics than the CDKN2A null expressing cell lines. In fact, there was a 2.6-fold increase in the IC50s for all of our chemotherapies tested if we overexpressed CDKN2A, suggesting that CDKN2A high expression may contribute to chemoresistance in Ewing sarcoma. But those are static lines, and we wanted to assess how chemotherapy could potentially impact the expression of P16, because in human tumors, 85% of those tumors do express CDKN2A and P16. What we found was that at baseline, the P16 expression in our endogenous lines was equal to the expression in our cell lines that didn't have CDKN2A in its genome at all. But when we treated those endogenous cell lines with low doses of chemotherapy, IC30s, the expression of P16 increased significantly and was not that different from our stable overexpression lines. So what this suggests is we now have an isogenic comparison. We can compare a stable low expression and a stable high expression state, and we can induce expression in our endogenous lines by treatment with chemotherapy. So we're continuing to work on this hypothesis. We're still in the very early stages of testing this at this point, but our current data suggests that CDKN2A, which codes for the protein P16, is likely a dynamic biomarker of clinical outcomes for these patients. In the early stage or in the late stage, both of those times where patients are not receiving chemotherapy, losing CDKN2A gives a survival advantage for the cells, so they grow faster and survival outcomes are worse. But during the brief point where we're giving chemotherapy, high expression of CDKN2A actually is beneficial for those cancers because they're able to avert chemotherapeutic cell death. Thank you all for having me here and for your attention. I do want to acknowledge the patients and families who've been so important to this work, students and trainees who have put so much time and effort into generating and communicating this data, and also to my mentors and collaborators, and then our funders as well. I'm happy to take questions. I know that was a lot of data and information, and I appreciate you all having me here and being willing to sit through a talk about sarcomas.